Welcome viewers, welcome to our Glory Globe TV. Malaria remains one of the world's deadliest diseases. Each year malaria infections results in hundreds or thousands of deaths, with the majority of fatalities occurring in the children under 5. The Center for Disease Control and Prevention recently announced that 5 cases of mosquito-borne malaria were detected in the United States, the first reported spread in the country in 2 decades. Fortunately, scientists are developing safe technologies to stop the transmission of malaria by genetically editing mosquitoes that spread the parasite that causes the disease. Researchers at the University of California, San Diego, led by Professor Omar Akbari laboratory, have imagined a new way to genetically suppress populations of Anopheles gambiae, the mosquito that's primarily spread malaria in Africa and contribute to economic poverty in affected region. The new system targets and kills female of A. gambiae populations since they bite and spread the disease. Publishing July 5th in the journal Science Adventures, first author Andres Milder, a podcaster scholar in the UC San Diego School of Biological Science, along with former master students and co-first authors James Pye and Rima Apte, created a system called Infecgenia, an acronym for Inherited Female Elimination by Genetically Encoded Nucleuses of Interpart Allies. The technique leverages the CRISPR technology to disrupt a gene known as female lace that controls sexual development in A. gambiae mosquitoes. Scientists at UC Berkeley and California Institute of Technology contributes to the research in effect. Infecgenia works by genetically encoding the 2 main elements of CRISPR within the African mosquitoes. Those includes a Cas9 nucleus, the molecular scissors that make it cut and guide RNA that redirects the system to the target through a technique developed in those mosquitoes. Akbari's lab, the genetically modified 2 mosquitoes families separately express Cas9 and the flea targeting guided RNA. We crossed them together in the offspring. It killed all the female mosquitoes, said Smilger. It was extraordinary. Meanwhile, a gambial male mosquito inherit infecgenia by the genetic edit doesn't impact the reproduction. They remain reproductively fit to mate and spread infecgenia. Parasite spread eventually is halted since female are removed and population reaches a reproductive dead end. The new system, and the authors note, circumvents certain genetic resistance roadblocks and control issued by faced by the system, such as gene dives since the Cas9 guide RNA. This technology has a potential to be safe, controllable and scalable solution to the world urgently needs to eliminate malaria once and for all, said Akbari, a professor in the Department of Cell and Development Biology. Now we need to transition our effort to seek social expense, regularity, use of authorization and funding opportunities to put this system to its ultimate test and suppressing wild malaria transmitting mosquitoes population. We are on the cusp of making a major impact in the world and we won't stop until that's achieved. Researchers note that the technology behind Infecgenia could be adapted to other species that spread deadly diseases, such as mosquitoes known to transmit dengue, chikungunya and yellow fever viruses. Bureau Report, Glory Globe TV.